Well, hello, everybody. Welcome to Pediatric Grand Rounds. I am going to turn this over to Dr. Mel Hyman to introduce our speaker for today.
Thank you, Dr. Hirsch. So it's my pleasure to introduce Dr. Julie Tomaino. She is a pediatric gastroenterologist, wonderful field, and Deputy Director for the Division of Gastroenterology at the Food and Drug Administration, where she's worked since 2012. She trained as a resident in pediatrics at Children's Hospital at Montefiore, Albert Einstein College of Medicine, and then as a fellow in pediatric gastroenterology at Mount Sinai Medical Center in New York, where she earned a Master of Science degree in clinical research as well. Upon completion of her fellowship, she joined the faculty in the Department of Gastroenterology, Hepatology, and Nutrition at the Ann and Robert Lurie Children's Hospital in Chicago, where she was until 2012 when she joined the FDA. Her interests have included inflammatory bowel disease, celiac disease, and gastroparesis. And she has represented the FDA's division on gastroenterology at multiple public workshops and conferences, and has authored publications and guidance documents in these areas. She has a strong desire to help facilitate drug development in the pediatric population and enjoys sharing her experiences at the FDA with students, residents, and fellows. Today, she will be speaking to us on an introduction to FDA, history, regulations, and clinical trial design. Dr. Tomaino, thank you.
Great, thank you so much for that nice introduction, and thank you all for having me. I'm going to switch to share my screen. All right, can you guys see my slides okay?
Yes.
Okay, wonderful. So again, good afternoon, everyone. Thank you for having me. I'm delighted to be here. As you heard, my name is Julie Tomaino. I'm the Deputy Director in the Division of Gastroenterology at the FDA, and I'm also a pediatric gastroenterologist. This is just our standard disclosure statement. And today I'm going to give you a brief overview of the FDA organizational structure, share some FDA history that you may not be aware of, review the basics of the regulations that guide our decision-making, and discuss some general considerations for trial design, and then focus on specific pediatric development aspects. This is FDA's mission statement. FDA is responsible for protecting the public health by assuring the safety, efficacy, and security of human and veterinary drugs, biologic products, and medical devices, and by ensuring the safety of the nation's food supply, cosmetics, and products that emit radiation. How we carry out this mission is rooted in the history of the laws and regulations that are the foundation for the framework that guides our decision-making. FDA approves drugs and biologic products, including therapeutic proteins, vaccines, and blood products. FDA does not develop or test products before approving them. Instead, experts review the results of the chemistry data, the animal data, and human clinical data to make a benefit-risk assessment. If FDA grants an approval, it means that the agency has determined that the benefit of the products outweigh the known risks for the intended use. And I'll explain more about the benefit-risk assessment a little bit later in my presentation. FDA is divided into several centers, as you can see here. There's the Center for Biologics Evaluation and Research, or CBER, that oversees blood products and vaccines. There's the Center for Devices and Radiologic Health, or CDRH, that oversees medical devices. There's the Center for Drug Evaluation and Research, or CDER, which provides regulatory oversight for drugs and therapeutic biologics. And the Division of Gastroenterology is located within CDER. And you can see that there's several other centers as well. Within CDER, there's several offices, and the Division of Gastroenterology is located in the Office of New Drugs, which provides regulatory oversight of new prescription drugs. There's a separate office, the Office of Generic Drugs, and they provide oversight of generic products. Within the Office of New Drugs, there are several other offices with review divisions under each one. And the box on the screen highlights the Office of Immunology and Inflammation, or OII, which oversees 5 clinical review divisions, including the Division of Gastroenterology, and 1 non-clinical review division. Alignment of these therapeutic areas facilitate interdivision collaboration within our office. For example, many products share indications across GI, rheumatology, allergy, or dermatology. So this promotes collaboration across our divisions and facilitates our review and discussions of safety issues that might affect programs as a whole or a drug class broadly. And here's the organization chart for our division. We have 4 therapeutically aligned clinical review teams, each led by a team leader, and 3 to 4 physicians and clinical analysts on each team. And our current division staff is located on this slide. And these are the indications that we most commonly cover within our division. It should be noted that the liver and nutrition related indications are managed by our sister division within the same office, the Division of Hepatology and Nutrition. Our division's commitments are closely aligned with CDER and OND missions, but with a narrower scope to focus on serving patients with GI disorders. We aim to facilitate development of safe and effective drugs intended to treat GI disorders as expeditiously as possible. And our ultimate stakeholders are the patients and their families. And we incorporate their experience to enhance understanding of each disease and to facilitate creative approaches to drug development. And we often collaborate with patient groups, the academic community, therapeutic developers, and other regulatory agencies to foster development of promising therapies that will benefit our patients. So now you have a general idea of the overall structure of the FDA. I'm going to switch gears a bit and provide a brief overview of the FDA history to give you an idea of how the regulatory framework was established over time. These are just a few of the early milestones. Starting back in 1820, 11 physicians met here in Washington, D.C. and established the U.S. Pharmacopoeia, which was the first compendium of standardized drugs for the United States. In 1848, the Drug Importation Act was passed by Congress, which required U.S. Customs and Service inspection to stop entry of adulterated drugs from overseas. And then in 1902, the Biologics Control Act was passed, which ensured the purity and safety of serums, vaccines, and similar products used to prevent or treat diseases in humans. Since FDA is a regulatory agency, the law is an important aspect of our history. And there are many, but I've selected just a few that play an important role in FDA's history. Prior to this Pure Food and Drug Act of 1906, which was signed by President Theodore Roosevelt, drugs were sold with misleading labels and undisclosed ingredients. This was one of a series of significant consumer protection laws enacted in the 20th century that eventually led to the creation of the FDA as we know it today. At this time, the FDA was called the Bureau of Chemistry. Now, this law was principally a truth in labeling law designed to raise standards in the food and drug industries. It also addressed addictive and dangerous ingredients in product labeling. Products needed to be labeled correctly, including contents and dosing. The 1912 amendment was prompted by an unfortunate situation with Mrs. Winslow's soothing syrup for teething and colicky babies, which was unlabeled and laced with morphine, and many infants died. Congress enacted the Shirley Amendment to overcome a ruling from the year prior in U.S. v. Johnson. Now, Johnson's mild combination treatment for cancer was an ineffective product with false therapeutic claims on its label. In U.S. v. Johnson, the Supreme Court ruled that the effectiveness claims were not within the scope of the 1906 Food and Drugs Act. This amendment brought therapeutic claims within the scope of the Act. However, the Bureau, which was the FDA at that time, was required to prove that those claims were false or fraudulent before they could be considered illegal. Later, in 1938, the 1906 Act was replaced by the Food, Drug, and Cosmetic Act under FDR. This act gave authority to the FDA to oversee the safety of food, drugs, medical devices, and cosmetics. This was prompted by the death of 71 adults and 34 children from September to October in 1937 and in 15 states from Virginia to California because of a product called elixir sulfanilamide. Sulfanilamide had been used safely to treat streptococcal infections for some time, but as a tablet and powder. A salesman for a pharmaceutical company noted the increased demand for a liquid form. The company lab found that the drug easily dissolved in diethylene glycol. Now, they tested this for fragrance and flavor. Apparently, it had a very nice raspberry flavor and appearance, but they did not test for toxicity. Now, you all know this is toxic if ingested. Because of the success in creating a liquid, they shipped the elixir 633 shipments all over the country. At this time, the 1906 law did not require safety studies. In early October 1937, the American Medical Association received reports from physicians that an unfamiliar sulfanilamide compound was responsible for a number of deaths. The AMA asked for samples of the drug, identified diethylene glycol as the toxic ingredient, and immediately issued a warning. A physician notified the FDA, and inspectors were sent to the company headquarters. They quickly communicated that the drug was lethal and all products should be returned immediately. State and local authorities joined the effort to make sure all of the product was retrieved. Now, at this time, safety was the primary overriding component of the law, even with the 1912 amendment, until another tragedy occurred, which prompted yet another change to the law. The 1962 amendment required drug makers to provide proof of not only safety, but also efficacy of their drugs before approval. This amendment was a response to the thalidomide tragedy in which thousands of children were born with birth defects outside of the US as a result of their mothers taking thalidomide for morning sickness during pregnancy. Thalidomide was available outside of the US, and it seemed like a straightforward application when it first came to the FDA. A new medical reviewer named Frances Kelsey identified a safety concern related to the birth defects and pushed for more information. Now, she could because of the 1938 Act related to safety of the drugs, but efficacy at that time was more anecdotal. Based on the safety findings, the product was not approved in the US and was eventually withdrawn from the market outside of the US. The company had distributed a small amount of product before the FDA review was complete, and there were 17 birth defects in the US population. However, it could have been much more devastating. Now, because of this, the 1962 amendment requires industry to prove effectiveness of drug products before the product goes to market. And this proof is based on adequate and well-controlled clinical trials conducted by qualified experts. You'll hear this again. It also incorporated informed consent into clinical trials, required FDA approval before the drug could be marketed in the US, and gave FDA control of prescription drug advertising. It also allowed FDA to conduct inspections of production facilities. Now, many years later, the Prescription Drug and User Fee Act was enacted in 1992. Now, this law authorized the agency to collect user fees from the pharmaceutical industry. And the impetus was to reduce the length of time between the submission of a marketing application and FDA's decision on approval or licensure. FDA has time clocks associated with various types of submissions that come to us. So, for example, a marketing application ranges from 6 to 8 months for a priority review and 10 to 12 months for a standard review. PDUFA must be reauthorized every 5 years and has been reauthorized 5 times, the last time in 2017. The next reauthorization will be September of 2022. President George W. Bush signed this law, the Food and Drug Administration Amendments Act, or FDAA. Among the many components of the law, the PDUFA User Fee Act and its device component, MDUFA, have been reauthorized and expanded. These programs ensure that FDA staff have the additional resources needed to conduct the complex and comprehensive assessment of the data necessary for new drugs and devices. Two other important laws that were signed years earlier but reauthorized and strengthened under this amendment include the Best Pharmaceutical for Children Act, or BPCA, and PREA, the Pediatric Research Equity Act. Both of these are designed to encourage more research and development of treatment for children. In addition, this amendment specifically provides FDA with the authority to require drug manufacturers to conduct post-market safety studies to assess possible serious risks associated with the drugs. And then the 21st Century Cures Act was signed in December 2016. This law builds on FDA's ongoing work to incorporate the perspective of patients into the development of drugs and biologic products and devices for FDA's decision-making process. Cures enhances our ability to modernize clinical trial design, including the use of real-world evidence and clinical outcome assessments, which help to speed development and review of novel medical products. All of these laws are codified within the Code of Federal Regulations, or the CFR, which describes the general and permanent rules published in the Federal Register by the executive departments and the agencies of the federal government. FDA decision-making is guided by its regulatory framework as well as precedents and policies. And Title 31 of the CFR is reserved for the rules and regulations of the FDA. So what do we really do with the FDA? In the context of marketing authorization of a drug, FDA's regulatory decision-making is based on a benefit-risk assessment. The benefit-risk assessment is the foundation for our regulatory review of drugs and biologic products. These assessments capture the agency's evidence, uncertainties, and reasoning used to arrive at its final determination for specific regulatory decisions. Additionally, they serve as a tool for communicating this information for those who wish to better understand FDA's thinking. We consider the evidence and uncertainties of several dimensions: analysis of the condition, current treatment options, if any are available, the benefit of the drug, the risks of the drug, and any risk management strategies. This approach provides a framework for assessing the available data and drawing conclusions on the main considerations that factor into our benefit-risk assessment. Now, there are usually some uncertainties about the product's benefit and risk to patients, and the body of evidence is inevitably incomplete, creating the need for scientific and regulatory judgment to determine whether the product's benefits outweigh the risks and whether additional measures are needed to address these uncertainties. And FDA has regulatory options to make decisions within the framework and address these uncertainties and to manage the risks. There are, of course, values and trade-offs between the benefit and the risks within the therapeutic context. Regulatory judgments consider what's important to patients as well as what is important to the public health, and we try to make the best decision for the public based on the data that are provided to us. The benefit risk assessment occurs throughout the product's life cycle, from development to approval and into the post-marketing setting. The challenges we face with any benefit risk assessment arise from the fact that this assessment is a qualitative approach based on quantification of asymmetric elements. And those are the benefits, the efficacy endpoints or outcomes, and the risks, the adverse events reported in the clinical trials and other sources. Our knowledge of the benefit risks evolves over time, as I said before, and we work in multidisciplinary teams to make this assessment, including physicians and clinical analysts with clinical and medical expertise, clinical pharmacologists, non-clinical toxicologists, statisticians, chemists, epidemiologists, and many others. The benefit risk assessment ties together all the dimensions in an overall analysis and provides a succinct explanation and rationale for our regulatory recommendations or decisions. Now, I've mentioned clinical benefit several times. So clinical benefit is established using evidence from clinical trials. And this is how we define substantial evidence in the law. If you think back to the slide with the picture of JFK and the Food, Drug, and Cosmetic Act. The substantial evidence of effectiveness is evidence consisting of adequate and well-controlled investigations by experts qualified by scientific training and experience to evaluate the effectiveness of the drug involved, and on the basis of which they could fairly and responsibly be concluded by such experts that the drug will have the effect it purports or is represented to have under the conditions of use. And this requires that studies are designed well enough to distinguish the effect of a drug from other influences, such as spontaneous change, placebo effect, or biased observation. While the statutory standard applies to all drugs, there's a wide range of uses for those drugs, which demand flexibility in applying the standard. FDA is required to exercise its scientific judgment to determine the kind and quantity of data that are required to provide for a particular drug to meet the regulatory standards. But flexibility does not mean that a marketing approval will be granted before demonstration of substantial evidence of effectiveness. So how does clinical benefit factor into drug development and trial design? Well, a key goal of any development program is, of course, to demonstrate the benefit of that therapy. Clinical benefit is a favorable effect on a meaningful aspect of how patients feel, function, or survive as a result of treatment. It should be meaningful, measurable, and interpretable. And then the observed benefit gets described in the labeling as a claim using words that represent the concepts measured, and they should also be meaningful and understandable to prescribers and patients. So as you saw written in the law, the first part of establishing substantial evidence of a clinical benefit is to conduct an adequate and well-controlled trial. So this means methods of assessment of the subject's response are well-defined and reliable. And then the protocol for the study and the report of the results should explain the variables measured, the methods of observation, and the criteria used to assess response. Specifically, clinical trials should have a clear statement of objectives, appropriate control for comparison, appropriate selection of patients with a disease or condition or who are at risk of the disease, baseline comparability, methods to minimize bias, appropriate methods for assessment of response, and then appropriate methods for analysis. The next few slides provide an overview of consideration for designing clinical trials intended for drug development and what we at FDA are often thinking about when we review trials. But even as you think about your own research, when you read a publication of a trial at a journal club, these concepts still apply. And so you want to design trials that can answer the scientific question and produce interpretable data. As you think about the overall design of the trial, it's important to understand the mechanism of action of the drug or the intervention. Is the drug intended as a chronic or episodic use or as a cure without the need for ongoing therapy? Is it intended as an adjunct to another therapy or as sole therapy? As you think about the enrollment criteria, identify your target population. The enrollment criteria should provide adequate assurance that the enrolled patients have documented disease and that the signs and symptoms are due to the underlying disease. And it's important to rule out other causes that might mimic other diseases. As you think about endpoints, what are you going to measure? Identify the signs and symptoms that, if improved, would constitute a benefit in that population. So what are the signs and symptoms experienced by patients that are intended to be treated with the drug? It's important to encourage patient involvement in the process and find out what is really meaningful to them. And what clinical improvements in signs and symptoms would convince you that patients have benefited from that treatment. Define the endpoint to reflect the outcome of interest that will be analyzed to address your research question. And then as you think about how you're going to measure the outcomes of interest, it's important to select or develop outcome assessments to measure that benefit. So how would you measure those core signs and symptoms experienced by the patients and demonstrate that benefit? How would you evaluate whether the underlying disease has improved or at least not worsened? And then it's important to establish a meaningful change that reflects that benefit. So what magnitude of change is considered meaningful to patients and will provide convincing evidence that the drug has shown a clear benefit? We consider all of these factors to determine whether a trial is adequately designed, whether the data support a meaningful benefit to patients, And we also need to decide whether the trial results are generalizable to the broader patient population that is likely to use the drug. So FDA has put forth previous recommendations to improve clinical trial diversity, including collection and analysis on data of racial and ethnic minorities, and measures that enhance diversity in clinical trials. This includes broadening the eligibility criteria when it's scientifically appropriate and to improve clinical trial participation. All of these groups have often been underrepresented in clinical trials, and FDA considers their enrollment to be a priority. FDA has, for some of these populations, already issued specific guidance to help ensure a diverse clinical trial participation and to generate sufficient data to characterize the safety and effectiveness across the entire population. And you can see a few of these guidance documents listed on this slide. How does all of this fit into pediatric drug development? Or how does pediatric drug development fit into all of this? Well, in the past, pediatrics has been more of an afterthought. And our goal is to encourage the planning of pediatric development programs much earlier in the process. The average time from adult approval to pediatric approval is about 8 to 10 years. And this results in off-label use, difficulties completing the pediatric studies, and lack of high-quality data to inform the use in pediatric patients. There have been several laws and regulations that encourage pediatric drug development, starting in 1997 up until 2017. And the two main pieces of legislation that promote evaluation of therapies in pediatric patients are PREA and BPCA. Think back to the slide I showed with President George W. Bush and FDA that reauthorized and strengthened these pieces of legislation. So pediatric studies are required with any new drug approval to which PREA applies: a new drug, a new indication, a new dosage form. But this requirement does not apply if the indication has received orphan designation, which are rare diseases affecting less than 200,000 US patients, or if there's a safety concern that precludes a pediatric study, then studies would not be required for that drug. However, even if a given indication has received orphan status, we still encourage the companies to conduct pediatric studies and work with them to conduct those studies intended to support labeling and ultimately approval to make the product available for pediatric patients. BPCA is a little bit different. This provides a financial incentive to companies to conduct pediatric studies. It also provides a mechanism for the National Institute of Health to identify drugs no longer under patent that need to be studied in children due to lack of dosing or lack of safety or efficacy data. Now, pediatric drug development is held to the same standard as adults for approval. You still have to demonstrate substantial evidence of effectiveness for the treatment of the proposed indication. You still need adequate safety information to be included in the application to allow for an appropriate benefit-risk analysis. The manufacturing still has to ensure product identity, strength, quality, purity. And ultimately, a successful pediatric program results in evidence-based labeling that adequately guides patients and prescribers on how to use the drug safely and effectively. So the approach to designing pediatric trials is generally the same as the considerations that I described earlier. But in addition, there's several things to think about and how the underlying pathophysiology is similar between the adults and children. Is it different for infants, children, and adolescents? Is the exposure response the same in pediatric patients and adults? Are the core signs and symptoms that define the disease the same between adults and children? Would that clinically meaningful outcome be the same or similar in adults and children? And what is the age range of pediatric patients who might benefit from the therapy? And then, of course, are there any uncertainties or limitations with the existing data or about the pediatric population? Now, we realize that conducting two large, fully powered trials in pediatric patients is very challenging in most situations. And so we exercise flexibility by relying on extrapolation of efficacy from adult trials to help facilitate drug development in pediatric patients. Extrapolation of efficacy is an approach to improve the efficiency and success of pediatric drug development. It relies on a series of evidence-based assumptions that reference adult trials or other pediatric trials and targets the pediatric populations that would be expected to have sufficiently similar disease course and response to therapy. Now, relatively lower prevalence or incidence of a condition in the pediatric versus the adult population alone does not justify the use of extrapolation. And there are several factors to consider for extrapolation. And the approach that one chooses depends on several things. Think about whether the disease is sufficiently similar between adults and pediatric patients. Is there a common pathophysiology or natural history? And is the response to treatment expected to be similar? If the evidence supports the disease similarity and the response similarity, then we need to think about what additional data are needed. Are there endpoints in the reference population that are relevant to the pediatric patients that can be used to predict efficacy? Now, on this slide refers to a PD or a biomarker endpoint, but a PD endpoint can be a clinical endpoint. So for example, in pediatric IBD trials, we use signs and symptoms assessed by the CVAI or the Mayo score and endoscopy as efficacy endpoints. The other factor to consider is whether there's similarity in the exposure response between adults and pediatric patients. And this is important to help select the dosing that you're going to evaluate in the clinical trials. Collecting robust data during the adult trials is critical to informing development in the pediatric patients. And sometimes the data are adequate to select an effective dose in adults, but leave some uncertainty for the range of dosing that would be needed in the pediatric population. Now, there are many techniques to make use of the optimal use of the available data. And we have to think about what additional information needs to be generated to inform the acceptability of extrapolation. All of these considerations are used to inform how the pediatric program is designed. Now you can see here that there's various approaches to designing the pediatric development program, and it's all based on the amount and confidence in the available data, most often from adult trials, but sometimes from other pediatric age groups. Now, out of all these possible approaches, approximately 60% of the pediatric programs still require at least one adequate and well-controlled efficacy trial. But you can see there are other possible approaches here. So you can have a controlled study without formal statistical power, a descriptive study, a small dose ranging study, efficacy safety studies, PK/PD studies, or PK and safety only. And again, what you choose really depends on the amount and confidence in the data that you already have. Now, I've shown various approaches to pediatric drug development, and it's important to remember that pediatric extrapolation applies to efficacy data only. Pediatric dosing and safety cannot be extrapolated, and sometimes pediatric PK studies are needed to inform the dose selection. Then the identified dose needs to be confirmed in a pediatric efficacy and safety trial, and safety needs to be evaluated in the target pediatric population. Now, as far as the dose selection, even with the pediatric PK studies, there's various approaches that one can use. And even modeling and simulation can explore a variety of pediatric dosing strategies to achieve a target range of exposures that then need to be confirmed in the pediatric study. So there's different ways that you can do this. So several take-home messages for you. FDA's mission is to protect the public health by assuring the safety, efficacy, and security of human drugs and biologic products. FDA assesses the benefits and the risks of the drugs and biologic products, and we make our benefit risk assessment. And a clinical benefit is a favorable effect on a meaningful aspect of how a patient feels, functions, or survives as a result of treatment. And it should be clinically meaningful, measurable, and interpretable. Regulatory flexibility is often needed to facilitate pediatric drug development by relying on the existing knowledge through extrapolation of efficacy, and frequent communication between FDA, industry sponsors, academic investigators and clinicians, and patients are critical to success. This is a picture of our FDA headquarters. Sits in White Oak, Maryland. And I've provided several resources here for you, some general information on FDA. You can access any of the approved product labels and our regulatory reviews if you're interested in reading any of those benefit risk assessments, seeing the rationale and the reasoning behind some of our regulatory decisions. Those can be accessed in the link in the second bullet. Our various guidance documents can be accessed, as well as some other resources here. I'd like to acknowledge just several people who I borrowed some slides from, adapted some of the slides. And then if you have any questions, I'd like to thank you all for listening.
Great, thank you. That was a really great introduction, and then follow on as far as the FDA and pediatrics. Really enjoyed that. I don't see any questions in the Q&A, but maybe I'll start off with one kind of general, which ties in a little bit with what's been going on today with the vaccine development. And that is the issue that we have in pediatrics, where it takes so long sometimes for drugs to get approved for children that it tends to put kids at disadvantage. And you pointed out that it can take 8 to 10 years longer for pediatric approval compared with adult approval. And so I'm just wondering, are there ways to get around that?
Yeah, that's a good question. And, sometimes it's one that we struggle with internally as well. A lot of it depends on, the disease that's being studied, the mechanism of action of the drug, any safety concerns, and if there are available data, to support studying pediatric patients. And, depending on the phase of development and what we know about the drug, pediatric studies might be able to be started earlier. Depending on the situation and the age of the patients, sometimes the pediatric patients can be, combined with the adult study. A lot of it depends on, trial feasibility and again, whether there are data to support that there would be a benefit to patients, and also understanding, the potential safety risks to the pediatric patients. So, again, it depends on the available data and what we know about that product, but there are ways to, sort of expedite the process a little bit and start studying the pediatric population a little bit early. Because really, as you said, the delay, results in lack of information, potentially off-label use. And so really having an understanding of the, effective and safe use, the doses that are appropriate for kids is really important.
Now, one of the problems we have with the off-label use is the insurance companies often refuse to cover it. So then we have to struggle with that as well. I'm sure you're aware. Does the FDA, so the FDA requires companies now to do pediatric trials, is that correct?
Yes.
Do they say that a company has met that requirement if they include, say, 14 to 18-year-olds in the adult trial?
So that's a good question, and it's one that I get pretty often. Again, it all depends. So there might be cases when, adolescent patients are included in the adult program, and we determine that there are sufficient data to really inform labeling in the population, including in the adolescent patients. So if there's enough data and we are able to label the product for the adolescent population, then we might say that requirement is fulfilled. Now, that doesn't mean that they wouldn't still be required to study younger pediatric patients if the disease occurs in the range of patients. So again, it depends on how much data are available, and whether we think that the product can be adequately labeled in that age group. But they likely would still be required to conduct studies in the younger patients.
Okay, thanks for that. So there's a question that relates to diversity and related. So the question is, how could you highlight some specific examples of how the FDA is taking a more equitable approach to enrolling a diverse pediatric population? I guess along with that is the whole, well, yeah, I'll leave that question.
Sure. Yeah, that's a great question. Whenever we look at the protocols that are coming in, one thing we think about is this trial going to generate data that we could generalize to the broader patient population, right? Because I think, what you're getting at in the question is that the trial population is sort of a very, specific group of patients that meet the eligibility criteria for that trial. But then once the drug gets out there on the market, it's available to the whole US, population. And so when we do review the trials and the enrollment criteria, we think about that, and we think about if there, would be knowledge gaps in the ability to use the product, safely in the broad population. And so we work with the companies to, make sure that the enrollment criteria, address that need, so that when we see the data, we can conclude that this product could really be used in the broader patient population.
Okay. Another thought is, obviously all these requirements for drug trials and requirements has a cost to that, which certainly adds to the cost of the development of these drugs for clinical use. How does the FDA take that into account, or does it, or is it strictly focused on safety and efficacy, and it's up to the pharmaceutical industry how the cost is dealt with?
Yeah, FDA, the cost of drugs doesn't fall under the FDA's purview. So that, that's not something that that we regulate or that we oversee. So when we review the protocols and the data, we are approaching it from a scientific and regulatory perspective. Although we, are aware of some of these challenges, we listen to the community, the patient community, the academic community. We've heard some of the concerns.
So there's a question. What are the considerations for preclinical models with respect to modeling pediatric PK/PD?
No, that's a good question. We have used preclinical models, and just to clarify the terminology, I think you're referring to animal models. That can be preclinical, non-clinical, but in the rare disease space where it's just not feasible, to conduct multiple pediatric studies, or maybe the disease occurs only in pediatric patients, we have used, preclinical models to help inform the dose selection and collect information to support, conducting the pediatric trial. So there is a role for it. Again, it depends on, the specific disease of interest, whether data could be collected in adults first or in older pediatric patients first. Sometimes the animal data are not as informative. It just depends on on what your intended use of that animal data is. But there is a role for it. It really it depends on the specific situation.
Another question I have for you is related to the orphan drug status. Seems to me that a lot of especially in an academic medical center, a lot of the children we deal with would fall into that category. How are they handled differently?
Yeah, so the orphan status is based on the whole population of people that have that disease. So it's not just the pediatric population. So for and this has actually changed over time. The approach has evolved over time as far as how we grant orphan designation. But if there were a population that falls into that category, again, if it occurs in both pediatrics and adults, although PREA wouldn't apply to require those studies, we do still often issue pediatric studies along with the adult approval. They fall into a different category. They're not required under PREA because of the orphan designation, but we do try to encourage, drug development in that population, knowing that there's probably a need there. I don't know if does that answer your question?
Yeah, kind of. I mean, I'm thinking more along the lines of, for example, genetic disorders or some of the more congenital anomalies where there may be medications that are potentially available but aren't being developed because there's so few patients.
Yeah, that's always a challenge. We actually do have a division at FDA that is specifically dedicated to rare diseases. And then, the genetic therapies are handled in the Center for Biologics and Evaluation Research, CBER. But there are regulatory approaches and flexibilities that can be applied, to collect information, even in those very rare diseases. It's a little bit more challenging and does require, some flexibility and creative thinking, even within the regulatory framework. But it could be done.
Yeah. What about, there's been discussion for many years that you may have even been involved with some of the nutritional therapies. Are those still outside the FDA's purview? Like I'm thinking of the enteral formulas and.
Yeah, it depends on what it is. So if it is intended as a nutritional formula, that's actually a separate center from where I sit. So in CDER, the Center for Drug Evaluation and Research, we regulate, therapies that are intended to, treat diseases. If it's intended as a nutritional supplement or therapy, so such as even infant formulas, that falls under CFSAN, which is the Center for Food Safety and Applied Nutrition. So it depends on the intended use of the product. If it's being used to treat or prevent a disease, then it would come to us, and it would be studied and regulated, the way that a drug would. But if it's intended for nutrition, then that is a little bit different. It could still fall under FDA purview, but it would be a separate center.
Yeah, the situations that I'm thinking that come to mind would be something like enteral nutrition for Crohn's disease or special formulas for renal failure, situations like that.
Mm-hmm. Yeah, it would depend on sort of the claim that the company is seeking, where it would go and if it would fall under our purview.
What about the the products that try to manipulate the gut microbiome? Are those are not part of FDA?
They can be. Sometimes we do see products that are aimed at the microbiome. Again, depending on the product and the intended claim, it might come to where my division sits in CDER, or it might go over to CBER. It depends on the product.
But probiotics in general, I believe, are not. Are those more considered nutritional products, or how are those viewed?
Yeah, I'm not 100% sure. I think those are more nutritional supplements. I don't think they come through us, but I'd have to double check on that.
Yeah, because some of them are being touted as being effective against different disorders, whether it's asthma or colic or, again, we go back to inflammatory bowel disease, something we know about. There's been a lot of discussion that may be changing the way that bacteria populate the intestinal tract makes a difference. Well, I don't have any other questions. I don't know, Rafi or Sandrine, if you have anything you want to add or.
Well, since we have a few more minutes, maybe I can ask Dr. Tomaino a question that's kind of outside your area of interest, I know, and it may be a little bit of an unfair question to ask, but I'm curious if you have any thoughts about or can share with us anything about what people are saying at the FDA regarding this Alzheimer's drug that was just approved that's generated so much reports in the media and controversy about whether or not the FDA should have approved it. Anything you can share with us about that?
Yeah, I'm aware of the drug. Unfortunately, I really am not the right person to comment on that. The way that our divisions are divided up. I'm in the GI division, and so we are aware of what the other one is doing, but I don't have enough firsthand knowledge of that product in that situation to really say too much about it. But I do understand what you're referring to, and I've also seen the publicly available information and the discussions surrounding that. But yeah, I'm not really the right person to comment on that.
Okay, well, that's what I assumed, but I thought I would ask. Good.
All right. I don't see any other questions, Rafi.
Okay, Sandrine, anything you want to add or?
No, I had some computer issues, so I'm going to actually have to watch the full talk on YouTube later. I missed a few pieces of it, but the parts that I thought was incredibly interesting. I do actually think that this issue that Dr. Hirsch brings up, it's the one thing that I actually was wondering, and if you addressed it in your talk, I'm sorry about that. You did at some point mention that the FDA doesn't get involved with price setting for drug companies. But I just wondered whether in the approval process, there are often ethical conversations about pricing of drugs and because you have these patients organizations that are powerful. You have the drug companies that have there's a lot of stakeholders, and it seems with this recent controversy that it's not always easy for the FDA to strictly stick to the scientific review and not get influenced by all these other stakeholders. Does that come up in your work in pediatrics?
I think it's something that all of us at FDA are aware of, and we hear a lot from our external stakeholders, and at least in my division, I mean, we value input from our patient groups, from the academic community. It is one of the challenges of working at a regulatory agency, and we do our best to focus on the science and try not to be influenced by some of these other factors. But we do listen to to the external stakeholders, and it's certainly, you're right. I mean, there's a lot of voices providing input. And that, it can be a challenge, in our role.
Thank you.
Okay, well, I guess we'll wrap it up then. And I want to thank you again, Dr. Tomaino, for great grand rounds, and thank everybody for participating, and wish everybody a good remainder of the week.
Thank you so much for having me. Have a great rest of the day.
Thank you.